we do filtration and how we form the urine, okay? So, we're just gonna lay it out now, and then we'll look at function in your information and stuff on Thursday. And then Tuesday, we'll come back to that as we talk about how certain parts of the nephron function for hydrogen and bicarbonate organization due to acid base. So the nephron is essentially a tube that has been punched in or kind of pushed in, a hollow tube, by the growth of our capillary bed, the glomerulus. So we have a visceral layer of the glomerulus, and we have a parietal layer. Now, this shows an artificial separation. In fact, they're closely invaginated. That visceral layer wraps around the capillaries, so they're intimately associated with the capillaries. There's not that much of a gap, but for a diagrammatic component, that's there. So we have filtration occurring. This is called the glomerular capsule or Bowman's capsule. I typically refer to it as the Bowman's capsule. The glomerulus itself are the blood vessels. Okay. Then we have the first portion of the nephron, and anything that's closer to the point of origin, if you recall, we use the term proximal, and it's highly bending around, hairpin turns and everything, so it becomes a proximal convoluted tubule. Then it dips down to form the nephron loop, and then it forms the distal convoluted tubule where it attaches to our collecting duct. All right? So I'm going to diagram that on this right-hand side of the board. And we have our afferent arteriole up here. So I'll put in the glomerular capillaries and draw the outer layer of the capsule. Okay, the visceral layer is going to be, as I said, intimately associated with those components. We'll come back and look at that in a little bit more detail. So this is known as the Bowman's capsule. Okay? For histology purposes, although we'll look at that more, or again more on Thursday, this is simple squamous epithelium. And that, remember we talked about our respiratory membrane when we were looking at the lungs? And the respiratory membrane was a sandwich. The filling was loose irregular connective tissue. And what were the slices of bread? One was the alveolar membrane, and the other was the capillary membrane. And those are what type of epithelia? Simple squamous, all right? So our filtration membrane is going to be the same thing without as much loose connective tissue. So we have the simple squamous epithelium of the capillary and the simple squamous epithelium of the Bowman's capsule. Instead of having loose irregular connective tissue, we have just the basement membrane is our filling. Okay? Then we have the proximal convoluted tubule. Proximal because it's the closest to our origin, which is the Bowman's capsule or glomerular capsule. And this is simple cuboidal epithelia. All right? I'll come back and do the histology. I'm going to raise the board over there and do the histology with you a little bit more. Then we start to form the nephron loop. So we have the thick segment, which is still simple cuboidal. And we have the thin segment, which is back to simple squamous. Okay? And notice I'm drawing a juxtamedullary nephron here, because its loop is going down into the medulla. And then we have a thick segment again. So the this direction. So this is known as the descending limb. Any guesses what this is called? The ascending limb. And the whole thing is the nephron loop. Or another alternate name is loop of Henle. Okay? You can use either one, loop of Henle or nephron loop. Yeah.
And you said all of that is simple squamous?
The skinny part.
The skinny, the thin?
Yeah, the thin segment is simple squamous and the thick is still simple cuboidal. And notice how this is coming over close to our capsule again. This is called the straight distal tubule. Not convoluted, but straight. Or distal straight tubule, if that's easier to remember. And then it attaches, it goes past the capsule, and it passes between the afferent and the efferent arteriole. And I know you can't erase like I can, so I'm going to redraw it over here again for you to follow through. This becomes very important in the regulation of the diameters of the afferent and efferent arterioles. All right, as soon as it does that, then it becomes convoluted again. And then it attaches to the collecting duct. All right, so this would be the distal convoluted tubule. All right, now that you have that in mind, I'm going to turn off the PowerPoint and redraw it over here with a little bit more of the histology associated with it. Okay, but we have Bowman's capsule, proximal convoluted tubule, nephron loop, distal convoluted tubule, to break it down into a simple component. All right? So, start up high here. And the epithelium is simple squamous. Then, this is simple cuboidal. And very, very tall microvilli. Remember how microvilli are usually short in comparison to cilia? But these are very long. Remember the stereocilia of the epididymis? Where they kind of tufted and looked like the tails of sperm, but they were just so long? So these are very, very long microvilli. Do microvilli move? No. What's the purpose of microvilli? Surface area. So you can see how important surface area is going to be, all right? So these cells are simple cuboidal. That's the way that moves. We draw an example of that cell. It has lots of folds in the bottom, increases surface area. Actually, let me draw that a little bit forward. I have room to put it in there. And then very tall microvilli. Okay. Now, if you had me for Bio 430 last spring, we talked about this as an example of a site of secondary active transport. We'll be talking about that on Thursday. Secondary active transport is a passive process, but it requires active transport for it to happen. Active transport requires ATP. So where on here would we put the mitochondria making the ATP? The microvilli are facing the filtrate, okay? So we're going to have sodium ions moving in from high to low and then get actively pumped out the bottom of the cell. We'll talk about that process on Thursday. So down at the bottom of the cell are where we find our active transport comes, and that's where we find our mitochondria, okay? So out here is where the mitochondria will be. So that's a characteristic cell of the proximal convoluted tubule. So I'm just going to put the initial here. All right. Then we have the straight segment. And then we see the thin segment. So, thick descending limb. So we look at it in cross section. This is simple squamous. Now it's down in the it's down in the medulla, so it's also where we see the vasorecta. So in cross section, we're going to see a simple squamous epithelia. And it could be a capillary, that could be the vasorecta, or it could be a thin descending limb. So we're going to know it's vasorecta when it has red blood cells in it. Okay? So this is the vasorecta. This would be the thin segment. Otherwise, they look the same. All right? Then we see the thick ascending becoming the straight, distal straight tubule. And you can't tell in a cross section whether it's ascending or descending, okay? cuboidal. Back to distal cuboidal, but this cell, simple cuboidal, doesn't have the tall microvilli. It's cuboidal, but it might have just a few kind of plasma membrane folds, and the nucleus is very close to the top. In fact, sometimes it can bulge the top. Okay? Same thing for the distal convoluted. The nucleus is going to be right up near the top. And we don't have microvilli. Okay?
Is the, so the, thick and thin is landing on a both simple
Uh-huh.
cuboidal? Oh, yeah. No. That's the big.
Right, and now this is going to add itself onto the collecting duct. Now, at first, the collecting duct starts out simple cuboidal, but we're just gonna consider it to be simple columnar. I won't ask you to identify it unless it's simple columnar. Okay? So notice I drew it a different color. This is our last structure to develop from the metanephric duct. So what I drew in green develops from the metanephric duct, what I drew in black develops from the mesonephric mesenchyme mesoderm, right? So, collecting duct, simple columnar. Okay, so that's the nephron. Everything in black is the nephron.
Are you gonna ask us for the thickness, for the ascending, descending, what are you gonna ask that we don't know the difference? Is it a thick or thin segment?
So the models, you can tell.
But not the
Histology, more time on the diagram? A lot more time? Okay, so in preparation for our functional lecture on Thursday, before we finish up by looking at the histology, our quiz on Thursday, because we have this on, then I'll show you some images of models. All right, so question number 6. When the efferent arterioles dilate, and those are still up on the board, does the glomerular filtration pressure increase or does it decrease? We're talking about within the glomerular capillaries. Is the pressure pushing water and small particles out of the capillary higher, or is the water pushing particles out across the capillary into the nephron capsule, the Mobius capsule, lower? So you think about the cars getting tunneled from 7 lanes to 2. All right, what's going to happen is you back up, but if you add 5 more lanes at the other end of that decrease, are the cars going to start going through faster at the other end, or are they going to go through slower? All right, because that's our purpose for having 2 sets of arterioles, to help us regulate pressure. Okay, so here's a diagram then of the nephron. You should be more familiar with some of these structures now that we've talked about them. So the nephron starts with the Mobius capsule here. And then we have the proximal convoluted tubule. Here's the thick straight. And we have the thin segment, and then the thick again. And it's straight until it comes between the afferent and efferent arterioles, and then becomes convoluted. Now, there are diagrams, artistic influence, or they don't understand the physiology, where they have a loop going the opposite direction, and this ascending and straight isn't anywhere close to where the blood supply is going into and coming out of the capsule. Okay, but it's necessary that it should. And so this opening of the capsule is called the vascular pole, where the afferent and efferent arterioles are passing into the, into the capsule itself, but into that depression. And where the proximal convoluted tubule is attached is called the urinary pole, even though it's filtrate that comes out, not urine. Okay, so where the afferent and efferent arterioles come to the glomerular capillaries, that's the vascular pole. And then here, where we are actually leaving to form the filtrate passes into the proximal convoluted tubule, is the urinary pole. You can see that on this model, and I have some images on canvas as well. And this is where we're going to start talking on Thursday as we talk about function. Okay? So here's the afferent arteriole. Here's the efferent arteriole. And here are the glomerular capillaries. The reason they're not red is because they're covered by the visceral layer of the capsule. This is what forms our filtration membrane. Simple squamous of the capillary and the visceral layer of our capsule. All right? So this would be the vascular pole, and here's the proximal convoluted tubule leading away. So my finger's now in the lumen of the urinary pole. All right? This structure right here is that portion of the distal straight tubule passing between the two. And this will become very important in regulation of blood pressure. All right? There's chemicals that are released, and it senses sodium ions and so on. That's what we'll talk about on Thursday. So this region here is this part of the distal straight tubule passing between the afferent and efferent tubules. So this would be the parietal layer of the capsule, and the visceral layer are specialized simple squamous cells attached to that. All right, so here's what it looks like histology-wise. Controls. So we have, this is what a lot of faculty use for teaching simple squamous epithelium in Bio 430. So we have the simple squamous epithelium out here of the parietal layer. And then all in here would be the simple squamous of both the capillaries and the visceral layer. We call the cells of the visceral layer podocytes because they have little feet-like processes that come and rest on the capillaries. These brighter pink cells that you see out here with all the, I call it foreign toilet paper look, tissue paper look, these are the proximal convoluted tubules. Okay, so we'll look at those next. So this irregular surface is because of those very long microvilli. So here are the nuclei, the folds on the basolateral membrane where we find the mitochondria, but even taller microvilli than are shown here. Okay. Now we're down in the nephron loop. So here would be the thin segments, and here you can see some of them have blood cells in them. Those would be the vasorectum, all right? But these right here don't have any blood cells. So those are probably the thin segment. Whether it's ascending or descending, I have no idea, okay? There's nothing significant about that structure. You can see a little bit better here. These are not good views of the collecting ducts because it's just not very good structure here. This is a better. So here, now we're down in the cortex, all right? And as you'll see up here, and we'll go over this again on Thursday, we never find the capsule in the medulla. So here we're in the, actually we're in the medulla. We never find the capsule in the medulla. We never find proximal distal and distal convoluted tubules in the medulla. We only find the nephron loop, all right? So here we're seeing some of the thick segment, all right, but it looks just like the proximal, but we're never going to see, if we look in the microscope, you'll never see this structure here. You see that structure? You know you're in the cortex. You'll never see the capsule in the medulla, all right? Now, this is small here, but this would be distal convoluted tubule. See how close the nuclei are to the surface, and it has a clean edge compared to that messy surface, all right? So here is distal. That has a clean edge. Notice how the nuclei are close to the surface. So this would be distal convoluted, and this would be proximal. And I'm not worried about identifying the straight segment because it looks the same. You can't tell if it's straight or convoluted. But this would be proximal with the long microvilli. This would be distal with the smooth edge and the nuclei near the surface, and we don't see the microvilli part. Okay. Again, this would be like the distal. See how the nuclei is clean and smooth compared to this one? So this would be, it's down in the cortex, it's down in the medulla, so it's thick, but distal convoluted is going to look exactly the same. Now look at these cells here. See how much taller they are? Those are the collecting ducts. Okay? And as we move towards the papillary ducts, they become even taller. So let's see the collecting ducts. What would you call this one? It's a distal. See how the nuclei are near the surface? Okay? Here, are all bulging up against the surface. So that's a characteristic of distal. Okay, so we'll stop there, and then on Thursday we'll come back and start with the capsule. I want to show you some models. Easier to do it from here than. So this is the model that I brought up to the front room to show you the placement of the diaphragm here. Here's the quadratus lumborum muscle and the psoas major. All right, and you're seeing the ureters coming down to the bladder. So here we have the cortex and the medulla. And then we have the minor calyx joining to form the major. Here we just have a single calyx, all right? Pelvis. What this is pointing to is the tip of one of the renal papillae coming out at you from the back. Here, this is showing the openings of the papillary ducts. Medullary pyramid with the papillary ducts going in there. So these would be segmental renal arteries, interlobar, arcuate, and cortical radiate or the interlobular. You can see the Bowman's capsules up here, but that's pretty small, okay? And it's showing the placement of some of the nephrons. A little bit clearer, but the same components. Now we have 4 of these now, 5. A couple of older ones and 1, 2, 3, 4, 5. So this is what I was showing. This is an older one. So you're seeing those same blood vessels. All right, this is a single pyramid with the cortex associated with it. So this would be an interlobar artery, arcuate, and cortical radiant. And you can see the afferent arterioles coming off, and then the efferent leaving, forming peritubular capillaries, and here's a vasorecta coming down. Okay, collecting duct, and then juxtamedullary nephron. Here is the afferent arteriole, efferent arteriole, and we'll talk about the special cells of the distal straight tubule, and the glomerular capillaries with a visceral layer, vascular pole, and urinary pole. All right, so the structure
How do you
is
know the
large.
difference between the afferent and efferent?
Afferent is larger in diameter.
Okay.
Efferent is smaller. And the specialized cells that are shown here are specialized smooth muscle cells that create an enzyme that helps create a cascade of activities for the release of ADH and aldosterone. But we'll talk about that. Keep saying what we're going to cover, but we'll cover that on Thursday. Okay. So again, that one larger, interlobar, arcuate, and interlobular or cortical radiant. Afferent arterioles coming off to form the glomerulus, efferent leaving to form the peritubular capillaries, and here the efferent is coming down to form the vasorecta. So it's a matter of sequence. So here's the capsule. Leaving the capsule is the proximal convoluted tubule. That then forms a descending limb, the thick and thin segment, the ascending limb, thick and thin and thick, which leads us to the distal straight and the distal convoluted. So it's kind of just learn it in sequence. The convoluted to, proximal convoluted to descending to ascending to distal convoluted, okay? And then the distal convoluted joins into the collecting duct. So here's what I went through, and this will make more sense after our functional discussion on Thursday. Now here's the terrifying model, all right, broken into 2 segments. So what you see here is what you see here. It's just been moved up so you can see the bottom part. So what I showed you first were collecting ducts. Down at the bottom, they join to form the papillary ducts, which empties the filtrate into the minor calyx. All right? So looking at the blood supply first, we have the arcuate artery. We have the cortical radiant, giving off afferent arterioles. Inside the capsule would be the glomerulus, which we can't see. Up here leaving would be the efferent. Notice how it's smaller in diameter. And that forms our peritubular capillaries. So this would be our peritubular capillary bed picking up what's been filtered out of the filtrate back into the blood. Okay? Down here, efferent is forming the vasorecta down the medulla. Peritubular capillaries are in the cortex. Vasorecta are in the medulla. All right, let's go to the nephron. So this is our capsule. Leaving the capsule in reddish brown here would be the proximal convoluted tubule. We follow that around, and that forms the descending limb. Then we have the ascending limb, forming the straight distal, and then distal convoluted, and then that joins the collecting duct. So all the different colors can be a little, what am I looking at? But if you start with the capsule and just follow it through, you can see that the purple is joining the collecting duct. That makes it distal convoluted, okay? The reddish brown is coming off the capsule. That makes it proximal convoluted. It's kind of like the heart again. You can figure out which chamber. Right atrium then flows into right ventricle, which forms the pulmonary trunk. That means the other atrium is the left, and it receives the pulmonary veins. So once you can have that figured out, you can make those associations. And here is the same thing but labeled, okay? So this should be up on canvas. And then ureter and the parts of the bladder that we'll talk about on Thursday. Thursday's content for Thursday is over, all right? Name the blood vessel, name the structure, everything in it. But no histology.